Thank you, Charlie. It's a pleasure to be back at Yale again and get a chance to talk about some of our latest work. See a lot of old friends and relatives. So I want to start by acknowledging a few recent papers. So one just accepted at Nature that will be the focus of the last part of the talk, led by Ben Hopkins and a team of researchers at Weill Cornell, and also Sid Mukherjee at Columbia, and his lab is collaborating on this work that really sets the story, the punchline of what I'll be talking about today. But the idea to test the effects of diet and intervention on insulin and glucose was really stimulated by work done by our Stand Up to Cancer team in women's cancers that now has come to an end, but involved collaboration with 7 institutions and a critical paper by Carlos Arteaga and Ingrid Meyer that was studying PI3 kinase aromatase inhibitors in breast cancer. That the data from this trial and following the patients led to a lot of the ideas that I'll talk about today. And then I also want to point out a paper from a former postdoc, Ning Wu, in collaboration with Tim McGraw, that revealed that TXNIP, thyrodoxine interacting protein, is probably the major, as a direct substrate of AKT, is probably the major regulator of glucose uptake into cancer cells. In the degradation of this protein is critical for activation of glucose uptake into most tumors, and also into muscle and other tissues, liver, et cetera, and mediates most of what insulin does with regard to glucose uptake. So a bit of background on PI3 kinase. We discovered this enzyme back in the 1980s, late 1980s, as an enzymatic activity that co-purified with a variety of oncoproteins. And when we first found the activity at that time, there were only 2 known species of phosphatidylinositol phosphorylated species that had been known. First discovered in 1949. Was PI4P and PI45P2. We assumed that this enzyme was phosphorylating the 4 position of the inositol ring because that was the only monophosphorylated form known to exist. But as we characterized the product, we discovered it was actually phosphorylating the 3 position. And that ultimately uncovered 5 additional phospholinositides, of which all possible combinations of the 3, 4, and 5 position can be phosphorylated. I'll focus on this triply phosphorylated lipid because it's the most critical lipid in response to what insulin does, and in fact most oncoproteins do in response to stimulation by growth factors. About 10 years after we discovered PI3 kinase, a tumor suppressor gene that had been identified by positional cloning by Ramon Parsons and others as being on human chromosome 10, hence the name phosphatase on P10 on 10. This enzyme was homologous to protein tyrosine phosphatases, and in fact does have a protein phosphatase activity. But Jack Dixon, who had worked on phosphatases much of his life, had realized that the turnover number as a protein phosphatase was unusually low, and sought other substrates and discovered it could dephosphorylate the 3 position of PIP3, and thereby was completely reversing what PI3 kinase was doing. So the observation then that P10, which is second only to P53, is the most frequently deleted tumor suppressor gene in PI3 kinase, which we now know from the TCGA effort is the most frequently mutated and activated oncogene, clearly puts this pathway of producing PIP3 absolutely critical for cell transformation. But in fact, if you go back through evolution and ask when did this lipid PIP3 first appear, and the enzyme, the PI3 kinase that generates it first appear, it appears in multicellular organisms. Yeast, for example, Saccharomyces cerevisiae, do not make PIP3, but flies and worms do. And so if we go back and look at the genes that we know mediate PI3 kinase activation in humans and mice and ask, what is conserved back to flies and worms? The answer is insulin/IGF-1 is the only activator you really see in that pathway for this, the enzyme that we'll talk about, the type 1 PI3 kinase. And IRS1 and FOXO and AKT are the pathway most highly conserved, shown genetically, to mediate responses in activation of this pathway in flies and worms. And those pathways end up controlling aging and also growth. So if we then focus on that pathway, insulin binds to the receptor. This phosphorylates, causes autophosphorylation receptor at sites that recruit the adapter proteins, IRS1 through 4. These proteins have, in the case of IRS1, 4 doublets of phosphotyrosine spaced about 20 amino acids apart. And these sites have the sequence context of tyrosine, methionine, X-methionine. We were able to show in the early 90s that that's precisely the optimal sequence for binding to the SH2 domains of PI3 kinase. And that spacing ends up to be perfect to grab both SH2 domains simultaneously. So as a consequence then, the regulatory subunit of PI3 kinase brings the catalytic subunit to the membrane, and this binding engages the SH2 domains and opens up the activity of the enzyme, allowing it to phosphorylate PI45P2 to make this triply phosphorylated lipid. I'm going into some detail on this, and I'll have another cartoon of how this works, because it turns out to be very important to understand the mutational events that cause cancer. Now, once PIP3 is generated, then we now know there are a number of proteins that have plexure homology domains, now about 25 proteins, that will bind to this lipid and get recruited to the plasma membrane and mediate downstream signaling. But again, if we go back through evolution and ask what's most highly conserved, it's the protein AKT and its upstream kinase PDK1, both of which have plexure homology domains that bind to PIP3. And this allows them to be recruited to the membrane, positions PDK1 at a site to phosphorylate AKT and thereby activate it. Once AKT is activated, it then phosphorylates a host of proteins. There are at least 200 substrates of AKT now claimed in the literature, and probably at least half of them are correct. For those of you who've tried to chase down what kinase phosphorylates a given site on a protein, you'll realize it's not a simple task. But these that I've highlighted here are protein substrates of AKT that are highly conserved back to flies and worms, and they have the sequence context, optimal sequence context for AKT, also highly conserved. And they can mediate most of the things that insulin does. So I should say that I was actually in the early 1980s, in fact, late 1970s, the major focus of my laboratory was not cancer, but rather diabetes, insulin signaling. At that time, we knew nothing about what insulin did, except that when you stimulate a liver with insulin, you would get glycogen storage, you would get suppression of gluconeogenesis, increased protein synthesis, and stimulate glucose uptake in muscle and fat. And we had no idea how any of that worked. In fact, insulin, when I started working on it, we didn't even know the insulin receptor was a tyrosine kinase. And so even after it was found to be a tyrosine kinase, it was still obscure what it phosphorylated because it seemed to only phosphorylate itself. But we now know that this cascade downstream of insulin signaling ultimately explains all of these targets, all of these responses. I pointed out TXNIP as a protein that internalizes and degrades the glucose transporter. We now know that AKT phosphorylation of TXNIP leads to its degradation, and that allows glucose transporters to stay on the cell surface. Phosphorylation of AS160 drives GLUT4 translocation to the plasma membrane. So insulin and through AKT regulates both accelerating transporter movement to the plasma membrane and decelerating internalization, thereby keeping high levels of glucose. And I should say that in cancer cells, I think this is the major way of explaining the Warburg effect, the fact that there's much higher glucose uptake in the cancer because of this phosphorylation of TXNIP. TSC2 is another target of AKT that we discovered about 15 years ago or so when Brendan Manning was in the laboratory in collaboration with John Blennis's lab. And this ultimately, you'll see in a couple of slides, explains how AKT regulates TOR signaling and protein synthesis. FOXO transcription factor suppresses gluconeogenesis. That's another direct target of AKT. And then glycogen synthase kinase 3, as the name implies, controls glycogen synthesis. So we can now explain how all of these things work downstream of insulin. And as long as P10 is around, you only get a signal for the appropriate period of time, and then the whole system resets itself. Now, I wanted to point out why these two tyrosine phosphorylation sites and how they're important not only in normal signaling, but also in oncogenic transformation. So this is a 10 angstrom resolution structure of PI3 kinase. And you can see the NSH2 domain of the regulatory subunit is trapped in a pocket. And this pocket involves multiple charge interactions, salt bonds that hold the NSH2 domain in place. And one of these salt bonds actually gets reversed in one of the most frequent mutations that occurs in cancer, a site on the so-called PIC domain. There's actually a third salt bond not shown here between the green C2 domain and NSH2 domain. That salt bond also gets mutated in cancer. And then there's a third salt bond that's near a C-terminal site that's also mutated. That site does not directly reverse the salt bond, but it probably affects it indirectly through a conformational change. So normally then the NSH2 domain is trapped in this pocket held through these salt bonds in an orientation that does not allow it to bind to phosphotyrosine. So this protein is sitting in the cytosol with one hand in the pocket and one hand available to bind to tyrosine phosphorylated proteins. The C-terminal SH2 domain is the only one free. So what happens when insulin phosphorylates IRS1 or another tyrosine kinase PDGF receptor phosphorylates itself, for example, where you have these two spaced phosphotyrosine sites, is that only the C-terminal SH2 domain can engage first. It's available. Once it engages, the proximity of the second SH2 domain and the 20 amino acid spacing of these two sites is appropriate to allow, whenever the NSH2 domain falls out of the pocket, to immediately be grabbed by the second phosphotyrosine and held in an open state where now the enzyme is active. So this association of the NSH2 domain not only prevents the NSH2 domain from binding, but it also prevents the catalytic activity from being functional. So there are two events here as a consequence of this. One is the localization of the plasma membrane where the substrate resides, and secondly, opening up the kinase domain so it can produce the product. There are other interactions. RAS will bind another domain, a RAS binding domain in the protein. It's not clear in the case of insulin signaling that RAS is particularly important, but in other signaling contexts, it does become important. So the two most frequent point mutations then, the E545K and the 10479 sites, are helping to open this up, and now the enzyme, instead of having one hand in the pocket, has both hands free and can grab onto tyrosine phosphorylated proteins much more readily. You still, however, have to have a tyrosine phosphorylated protein for PI3 kinase to get to the membrane. So this is not like a KRAS mutation or EGF receptor mutation where the mutation allows the enzyme to be active independent of an upstream signal. This sensitizes PI3 kinase to an upstream signal, but you still need the upstream signal to really get significant production of this product, PIP3. So one reason we know that this model is correct is that there's a fortunately an infrequent event in which a child picks up a mutation in the P85 subunit. This probably happens in in the egg or sperm because it's not inherited. And the children end up appearing initially to have type 1 diabetes, and that these two children are the same age. This person has this short syndrome, and this disease has been known for some time. It was obscure what was going on. Insulin was being produced at very high levels, so if you measure serum insulin, you get screaming high levels of serum insulin in these patients, and yet they failed to thrive. Insulin receptor is highly tyrosine phosphorylated. So whole exome sequencing was done, and these children turn out to have a mutation in the C-terminal SH2 domain. So that first ability to grab onto the iris proteins is blocked by this mutation, and as a consequence, you cannot trigger PI3 kinase at all. Now, there's another mutation also only discovered, and this mutation, I should say, was only discovered about 4 years ago. There's another mutation discovered about 6 years ago in a disease called localized hypertrophy. And these are actually called a series of different diseases. Depending on where the hypertrophy is, it's given a different name. Can even be in the brain of neuronal cells at hypergrowth. And what happens is during embryonic development, a single cell picks up a mutation. It's a mosaicism. And every cell in the lineage of this now has PI3 kinase hyperactivated due to a mutation in PIK3CA. In fact, the same two mutations that occur in cancer explain this disease. It's not a true cancer because you still need a stem cell. You need a mutation to maintain stemness to get a cancer. But you have this hypergrowth phenotype, very similar to what we see if we introduce those mutations into tissues in a mouse. So in fact, if you have a large area of the tissue that has this mutation, the patient is actually hypoglycemic because so much glucose is taken up into the affected tissue that the rest of the body is deprived. If it's very small, you don't have that effect. So this clearly shows that the ability to take up glucose into tissue and for the tissue to grow is enhanced by these mutations and sensitize the tissue to insulin. So as long as everything works fine, then PIP3 is only produced in tissues when a growth factor tells it to be produced, and P10 will shut this back off again. And as long as every tissue only gets the appropriate amount of glucose during growth as is needed to form a normal organism, from a fly to a worm to a human, everything is perfectly balanced. You get a perfectly developed human being. If some tissue has a mutation in PI3 kinase that allows it to get more glucose than the rest and to have a more driven growth, then you get these hypergrowth phenotypes. Also P10, activating mutations in PIK3CA, overexpression of growth factor receptors, mutations of growth factor receptors, or overexpression of growth factors themselves can all drive this pathway to give these phenotypes that can ultimately result in cancer. Now, if we ask in more detail what is actually driving the growth, it turns out to be quite complicated. I don't have time in this talk to go through it all. This network was figured out, took several years to do it. We started out at the top trying to find out what tyrosine kinases were doing to signal in downstream, found PI3 kinase. At those days, I was playing basketball with John Blennis, who was a postdoc in Ray Erickson's laboratory when I was on the faculty at Harvard in Cambridge. And John was working, he had made this observation that the S6 subunit ribosome is highly phosphorylated in Sark-transformed cells. So he started out with phospho S6, tried to work his way backwards, and I was here working forward. It was actually a collaboration between John's lab and mine that led to the observation that AKT phosphorylates TSC2 to control a RAS-like protein called Reb that ultimately turns on the mTOR signaling pathway. And it's remarkable how fast science can move when you collaborate. It only took us 25 years to join hands. The entire railway across the country was done in far less time. So, but this network, then we know, is highly conserved from flies and worms to humans, and it senses not only whether a growth factor is telling the cell to grow, but whether or not there's enough energy for the cell to grow, and whether or not there are sufficient amino acids. And so the complexity of this has all been worked out in a number of labs. The actual discovery of TOR, target of rapamycin, was made by Michael Hall when he looked for rapamycin resistance in yeast. And that got him the Lasker Award last year. So I'm going to the reason I showed this slide is we use quite often in the laboratory and also now in the clinic phosphorylation of AKT by PDK1 or phosphorylation of S6 as convenient markers that either the PI3 kinase pathway is turned on or the TOR pathway is turned on. And so as I start showing some of the data with our mouse models, keep in mind that these are the probes we're using to try to tease out what's going on in vivo. So I mentioned that PI3 kinase is one of the most frequently mutated oncogenes. In fact, it slightly edges out KRAS, but that's probably because there have been more endometrial cancers sequenced than lung cancers. So Roy would probably argue with me if we counted all the lung cancers, KRAS might be higher. If you ask how often, do you either get a PIK3CA mutation or an amplification or loss of P10, either single allele light blue or double allele loss of P10, or inactivating mutation in P10, then this is what you see. And most of the major cancers that are responsible for deaths in America have a prominent loss of at least one of these two genes, or a loss of this gene or activation of that gene. If you look at all the nodes in that pathway, everything in red is an oncogene, everything in blue is a tumor suppressor gene, and you compare all of these, something like 95% of glioblastomas has at least one of these genes mutated. About more than 90% of endometrial cancers have at least one of these genes mutated. In many cases, you see 2 or 3 of these genes mutated in the same cell in an endometrial cancer. So the pathway really does dominate. If you just look at PIK3CA alone, it's women's cancers that seem to have the most frequent mutations in PIK3CA, and that's why we received this grant from Stanford Cancer to focus on women's cancers and try to understand how to better use PI3 kinase inhibitors to treat women's cancers. So this is an example of a patient who went on one of our early trials of our Stanford Cancer team. And this was a patient of Jose Baselga's group, at that time at Barcelona, before he moved to Mass General and before he moved to Sloan Kettering, all of which he did while still on our team. This patient had a PIK3CA mutation, 1047 actually. She had failed. She also had HER2 amplification. She'd gone on standard of care, aromatase inhibitor. She was ER positive, ER/PR positive. Also, she'd gone on Herceptin, ultimately lapatinib, progressed on that, and then finally ended up in our trial with progressive disease. You can see the liver met here by FDG PET, and she went on a combination of the BYL719, alpha-specific PI3 kinase inhibitor from Novartis, and letrozole, aromatase inhibitor. And after the first month on the drug, these METs were metabolically silent. So she had turned off glucose. The drug had turned off glucose uptake in the tumor. But if imaged in other ways, these METs were still there. They had not shrunk. They were still there, still the same size. They just weren't taking up glucose anymore. But after 3 months, they began to shrink. And by a year, she essentially had no visible disease. Now, unfortunately, by 2 years, she progressed and ultimately had to go off therapy. But it was this kind of result that got us very excited that there's something here happening in the clinic that we can do something about. And of course, turning off glucose uptake makes sense given the pathway I just told you about. So this is the results from this early, one of the phase 2 trials that we did with letrozole plus BYL719. The upper half are patients who did not have PIK3CA mutations. All of these are ER positive breast cancer patients. The upper ones did not have mutations. The lower patients did have mutations. And you can see more responders in the bottom half, but a lot of patients who had little or no response, and some pretty dramatic responders in the arm that did not have PIK3CA mutations. So we took a deep dive on this sequencing these tumors to try to see if we could understand why are these patients with mutations not responding, and why are these patients who don't have mutations responding. And the bottom line is we could not come up with a rationale. We looked at, do they have KRAS? Do they have P10 loss? You know, what else is going on? There was no logical explanation that we could come up with to explain why we didn't get such a strong correlation. This is very different from, Gleevec and BCR-ABL or gefitinib and EGF receptor, et cetera, or ALK and ALK inhibitor. So why can we not predict the responders better? And I should say this failure to be able to predict responders is why these drugs are still not approved. They're in approval trials and they may in fact get approved, but it's been a struggle because of this kind of difficulty in predicting responders. Here's another trial. This one was led by Jose Baselga, and everything with an asterisk has a PIK3CA mutation, and it looks kind of encouraging that these biggest responders here did have PIK3CA mutations. By the way, this is a different drug. This is GDC-032 from Genentech that hits the alpha and the delta and gamma, but not the beta. So this looks a little better. And this drug, there's some evidence, preferentially hits mutant over wild type, although it's fairly subtle. And that may be why you get a little better correlation with mutational events. But here are mutated patients who had immediately progressed, and a lot of stable ones that did or did not, and some responses that stable disease that didn't have mutations either. So again, not a perfect home run in predicting responders. But the fact that this behaved a little better gave me more support for the idea that it was progressing my mind during these therapies. And the idea was, I should say one additional thing, if we look at FDG PET uptake, the responders tended to have, in the first cycle, an FDG PET response, and the ones that did not respond tended not to have much response. So there's a sort of a correlation between what you see in the first cycle and whether or not they ultimately respond. But what occurred to me was that maybe this failure to get a perfect correlation between mutations and responses to the drugs had to do with what the patients ate, or whether or not they exercised, or in general, what their ambient insulin was. Because keep in mind that the best possible way to activate PI3 kinase is with insulin. And we were paying very little attention to what the insulin level was in the serum of the patients. And when the patients first enrolled, we paid a lot of attention. Certainly measured glucose levels, because you'd expect if you turn off PI3 kinase, the one that mediates insulin responses, you should get a dramatic increase in glucose. So we'll get into details on this, but this will raise the possibility that we should pay better attention to what's going on, not only with glucose in the patients, but also with insulin. So for those of you who don't think about insulin and glucose and insulin resistance all the time, here's a very simple version of it, in which you eat your pasta. Some of you may even be brave enough to add sugar to something you drink. And the consequence will be your glucose will go up. It will go up slower if you add pasta than if you have sugar. But in any event, it will go up. The glucose will stimulate the pancreas to release insulin. Insulin will then come back and tell the liver to quit making glucose because there's already high glucose in the bloodstream. Store that glucose, whatever glucose gets into the liver is glycogen. And I left out the muscle, but it will also tell the muscle to take, and the fat to take up glucose and store as glycogen or fat. So this is, of course, how everything works to make sure our glucose comes back to homeostasis as quickly as possible. If you happen to have a microtumor growing in the body, in this case the tumor looks bigger than the liver, but no matter how big the tumor is, if it has the insulin receptor, and I should say that these tumors that we're looking at, endometrial tumors, breast tumors, all of them that have those PIK3CA mutations, all have very high levels of insulin receptor, almost invariably higher than the tissue from which they evolved. So this insulin elevation will stimulate glucose uptake into the tumor as well. And if the tumor happens to have a PIK3CA mutation, then it will be hypersensitized to take up glucose. It will take up more glucose than the liver or the muscle, just like that localized hypertrophy does. If the patient happens to be insulin resistant, that means that the liver and muscle and fat are not properly responding to ambient insulin, and so the pancreas has to churn out much more insulin in order to stimulate glucose uptake and storage of glycogen and suppress gluconeogenesis. And that very high level of insulin will even better activate PI3 kinase in the tumor because the tumor is not insulin resistant, it's hyperinsulin sensitive, particularly if it has those mutations and if it loses P10. So, and if, of course, you're on a high glycemic index diet, then you've got a problem with insulin all the time. So this was what worried me, that it's this ambient insulin that's really the problem that may explain why everybody doesn't respond. So we decided to go in and measure more carefully glucose and insulin levels. Now, every patient who goes on a PI3 kinase inhibitor trial has an endocrine consultation. The glucose is monitored over the first 4 hours of taking the drug, the oral drugs. These are oral drugs. And you see something that looks just like this. These are actually mice, but in fact, the patient data is virtually identical to this. That within 10, 15 minutes, you begin to see glucose rise as you're turning off everything insulin does in the body. And the glucose continues to rise. I should say, if a patient is insulin resistant, they're immediately excluded from the trial. Type 2 diabetics are excluded. So everyone has to be non-diagnosed. But about a third of the patients that went on our trial were discovered to be insulin resistant as a consequence of trying to take the drug, because the glucose would go up so high that the endocrinologist would take them off the trial. So many, many people in America don't know they're insulin resistant. The good way to find out is to try to take one of these drugs. So anyway, this didn't bother me. This is exactly what I expected to see. In fact, one company came to me and said, please work with us. We have a PI3 kinase alpha inhibitor that does not cause elevation in glucose. I said, well, it doesn't elevate glucose. It's not hitting PI3 kinase, so why should we use it? So this was thought to be a good thing. But in fact, what bothered me was not that the glucose went up, but the fact that the glucose leveled off and started going down, even though these drugs have PK/PD characteristics that would say that at this period of time, the glucose level in the bloodstream is still pretty high enough to turn off PI3 kinase. So why did the glucose level come back down? That was the part that bothered me. In fact, that the only logical explanation for this is the insulin level must be extremely high to be able to counteract the effect of the drug on PI3 kinase in liver and muscle. And in fact, that turns out to be the case. These 3 drugs, all of them hit the alpha isoform. This one is the pan-PI3 kinase inhibitors. These 2 I already described to you. So they all 3 cause insulin levels to go up very high. And in fact, the doses of insulin you would see in the serum hit, if you gave a patient something who was not on the PI3 kinase that elevated their insulin that much, they would be dead in about 5 minutes. So this is extremely high levels of insulin we're seeing. So we ask what happens then if we take a tumor ex vivo and give it the sort of levels of insulin that we see in the serum of the patients and in the mice after about 15 minutes of dosing, which is around 10 nanograms per mil. So this is an endometrial cancer. It has a PIK3CA mutation. Endometrial cancers in general have high levels of insulin receptor. There are two different organoids here. These are from 2 different patients. We grew these organoids ex vivo and then gave them the drug BKM120, the pan-PI3 kinase inhibitor, titrated. And you can see that these tumors responded extremely well to the PI3 kinase inhibitor. The therapeutic doses are in the micromolar range, and by the time we get to micromolar, we essentially killed every cell in those organoids. So that's encouraging, but what happens if we add 10 nanograms per mil of insulin, the concentration you would see within about 20 minutes of taking the PI3 kinase inhibitor, and now more than half the cells are surviving within 10 nanograms per mil. And if you keep titrating it up, you can essentially save the entire organoid at the doses of insulin that are achieved in vivo. So this is telling us that we're probably, even though we're giving these oral drugs twice a day, and PK/PD says that they're under conditions almost all the time where they should be able to turn off PI3 kinase. In fact, it's only the first 15 minutes that the drugs have a chance to kill a cell. After that, the insulin levels are high enough to rescue the tumor. So can we replicate that in another model? So these are actually mouse pancreatic cancer cells. They're not PI3K mutant. It's the KRAS P53 mutated pancreatic cancer. 2 different cell lines derived from these tumors. We've been working with these for about 15 years in a collaboration with Ron DePino's group. And what we find is that if you give insulin to these ex vivo, you see that there's already a basal AKT activation. When you give insulin, it goes up even higher, quite high. So these have insulin receptor. All pancreatic tumors have very high insulin receptor. If you give PI3 kinase inhibitor without giving insulin, you can shut that off almost completely. Shuts off phospholipase 6, downstream signaling to press 40. But if you give insulin at the 10 nanograms per mil dose that you would see 20 minutes after the patient took the drug, now it keeps AKT active, in fact, higher than it was before giving the PI3 kinase inhibitor, before giving insulin. You keep AKT activated, you keep phospholipase 6 activated, press 40 is substrate of AKT, now phosphorylated. So that dose of insulin that you're seeing in the serum is enough to reactivate the tumor in both cell lines. So how can we keep the insulin level low? It's this insulin rebound we're arguing that's rescuing the tumor from being killed by the PI3 kinase inhibitor. How can we keep it low? One of my brothers is an endocrinologist, so I spent a lot of time talking to him about this. And so here's what one uses in the clinic if you want to keep insulin low. Of course, a lot of cases patients have to take insulin. This is something we avoid. It's written into our trials. If the patient has to take insulin to manage the glucose, they have to go off the trial. We anticipated this could be a problem. All the patients on our trials go on metformin, but as you'll see in a moment, metformin is not very effective at keeping the insulin down compared to other approaches. So the two approaches that I'll show you are most effective are a ketogenic diet and a sodium glucose cotransporter inhibitor. And here's the data on this. So these again are mice. We give them the PI3 kinase inhibitor at time 0. Glucose levels start climbing. And you can see that if you give metformin, you can somewhat slow down the peak, but ultimately it's not very effective. Ketogenic diet, this ketogenic diet was started only 12 hours before. We did not do a week run-in, but it clearly is more effective. If we did a week run-in, it almost flattens that line. And then a sodium glucose cotransporter inhibitor is more effective than metformin. And if you look at the serum as measured by C-peptide, the serum insulin levels as measured by C-peptide, the sodium glucose transporter and ketogenic diet are far better than metformin in keeping the insulin low during this therapy. So we go back to the tumors. Now these are in vivo with the tumor. This tumor is, again, an endometrial cancer tumor that has PIK3CA mutation. In fact, that same organoid that I showed you earlier is now transplanted subcutaneously in the mouse. And if we just look at insulin activity, insulin receptor activity as measured by tyrosine phosphorylation of the insulin receptor, there's an antibody that recognizes the autophosphorylation sites. You can see brown stain here indicating that these tumor cells have some basal tyrosine phosphorylation of the insulin receptor. If we now give the PI3 kinase inhibitor, you can see what happens in the tumor. The brown indicates the insulin receptor is dramatically tyrosine phosphorylated in this tumor. If you put the mouse on a ketogenic diet, you now see most of that brown stain goes away. And if ketogenic diet at the bottom, most of the brown stain goes away, except for the endothelial cells, which are still a bit brown. So even in the presence of BKM120, you've eliminated the ability of insulin receptor to autophosphorylate. So this keeps the insulin receptor low enough that you don't activate insulin receptor in the tumor. If you look at phospholipase 6, you see a similar result. In the absence of anything at all, you see, but presence of BKM120, but no other intervention, you see high phospholipase 6, indicating again you're getting this rebound and actually activating phosphorylation of S6 by giving the PI3 kinase inhibitor to the mouse. Metformin doesn't really help that. Sodium glucose cotransporter inhibitor helps a lot, but the ketogenic diet wins in blocking phosphorylation of S6. So our two reporters that the pathway are activated, both can be shut off if we put on a ketogenic diet. So does that do any good? So here's the punchline of the talk. that, yes, it does do a lot of good. So this is a KRAS PP3 pancreatic cancer, no PIK3CA mutation. PI3 kinase inhibitor by itself has little effect. Sodium glucose cotransporter inhibitor on top of a PI3 kinase inhibitor, BYL719, dramatically improves responses, but the best responses with the ketogenic diet, again, run in for at least a week before starting the oral therapy. And now you get shrinkage of the tumor. Here's another, again, another allograft. So in this case, the tumor is put straight into the pancreas. So we're putting a mouse pancreatic tumor into a mouse pancreas of a littermate and expanding it into multiple mice. They're all then treated by a variety of therapies. The therapy that clearly works the best is the combination, again, the ketogenic diet plus BYL719, where in this case the tumor completely goes away. And we then ask what happens if instead of giving the ketogenic diet, we just use a hairpin knockdown, an shRNA doxy-inducible knockdown of insulin receptor in the tumor. And that's almost as effective now in combination with the PI3 kinase inhibitor as the ketogenic diet. So this says that if you can turn off insulin receptor in the tumor, you can get the same effect as a ketogenic diet, almost. Ketogenic diet still wins because we don't completely knock out insulin receptor by this approach. So this very strongly argues it's through the insulin receptor that this rescue of the cells is working. But this is the experiment I like best. And you can see that there's one color here that's different from all the other colors. So there were a lot of different variations on this experiment. And the brown is the ketogenic diet plus BYL719. This is a triple negative breast cancer with a PIK3CA mutation. And you can see this tumor completely goes away if we give BYL and a ketogenic diet. BYL alone does nothing for this tumor. And if you add back now the ketogenic diet plus BYL, the same as the brown, except now you inject insulin into the peritoneal cavity at the same time you give the PI3 kinase inhibitor, you completely eliminate the ability of the ketogenic diet plus BYL to shrink the tumor. So I think this says without any doubt, it's this elevation in insulin and activation of insulin receptor in the tumor is why these drugs are not working. The insulin level going up is turning on the PI3 kinase pathway in the tumor, overriding the ability of the drug to inhibit. This is yet another model, same result, ketogenic diet. Here's a Kaplan-Meier curve. Ketogenic diet keeps the mice alive on the PI3 kinase inhibitor. Otherwise, they die. So this is then the punchline, and I don't think I need to go through all these statements. It's pretty obvious that the reason these drugs are failing or being difficult to approve is because of this insulin overshoot. It's something we can readily control if we keep patients on a ketogenic diet. A sodium glucose cotransporter inhibitor is almost as good. We could probably do a combination of a sodium glucose cotransporter inhibitor plus a modified ketogenic diet that's a little more palatable to people. But we are now working with Bayer. Bayer has a PI3 kinase alpha delta inhibitor that's approved for follicular lymphoma. There it's primarily working by blocking the delta inhibitor, and therefore delta inhibitor does not mediate insulin responses, so you don't get that rebound effect so much in lymphoma. But Bayer is actually very excited about doing the ketogenic diet trial with that drug, which also hits the alpha isoform in the context of both solid tumors and liquid tumors. I left out the data, but we have data with Sid Mukherjee's laboratory that AMLs also respond dramatically to the ketogenic diet plus PI3 kinase alpha inhibitors as well. So with that, I'm glad to take any questions.